Yeah.
Okay. Good morning, everybody. Before I get started with our lesson, I wanted to remind you that if you are missing any assignments, that you still can submit. So I don't want you to miss credit for those assignments. I know Miss Emily and Miss Athena have mentioned before how important it is that you're completing the work, so you do get your stipend. And if you're having trouble with anything or any questions, please reach out to me or one of the TCs. I have office hours tomorrow, so we're here to help. So even if it's not correct, like your answers, that's fine. We just want to see that you're trying and doing the work and uploading your videos or pictures that we want to see. So, Emily and Athena, you want to kind of add to that, please?
Go ahead. Also, Amy sent us a list of students who haven't done anything in any of their classes. So, we'll be reaching out to you to figure out why you haven't done any work. Hey, Lamar.
Okay. So this week is kind of a segue into basically how your body is working, but you're going to be doing a lab on osmosis. And it's one of my favorite labs to do. So I'm really excited, and I hope you guys have a really good time with it. I know the past 2 weeks we've focused on chemical reactions and why certain things are reacting together and what the end result is. And even with this lab this week, it kind of goes off of that idea. So in week 3, the module, I only published right now the slideshow that I'm about to walk you through because it is a lot of information. I know. You don't really need to know all of it. I really want you to get the concept, though. That is my goal, that you understand the main concept, and that after doing this lab at the end of the week, you're kind of like, you have that aha moment, like, okay, like I kind of get that. So that's kind of what I'm looking for. So I'm going to share my screen and pull up the slideshow that I posted for you. My computer is not cooperating right now. There we go. All right, so if you haven't gone through the slideshow yet, it's okay. I'm going to walk you through it now, but I'm going to kind of speed through a few things. So if you want to go back to it, like I said, it is posted. So this whole show that I'm about to walk you through is on cell transport, how things are getting in and how they're getting out of the cell. And that might sound or seem like a pretty easy concept, but your cells in your body or any living organism are working pretty hard to keep you alive. And it's kind of crazy just kind of thinking about it if you're putting it into perspective, like how much your body is doing without you even knowing it. So by the end of this lesson, you should be able to explain the difference between active and passive transport. You should be able to explain the difference between diffusion and osmosis. You should be able to list 2 types of passive transport, give examples of diffusion and osmosis in any living organism, and also explain why our cells are actively moving the things we need and the things we don't need out of our cells. So I want to first explain to you the difference between what passive and active transport are. So you may have never even heard of that before, but when you start thinking about those words, what the word passive and active mean, and then the word transport, it should start to make a little sense. So if you've ever heard of somebody that's being passive, like a passive personality, that just means when somebody is kind of like laid back, they kind of let things slide, they don't really react in a major way, they're not aggressive. So when you think of passive, think about something that's not using a lot of energy or any energy for that matter. And then in terms of active, think about when you're being active, when you're exerting a lot of energy to do something. Like when I think of being active, I think of working out or going for a jog, riding my bike, doing something that I'm exerting a lot of energy. And then transport, like transporting something, whether it's going from A to B. When you get in the car, or if you're on the train, or if you're on your bike and you're trying to get somewhere, that's when you're transporting something. So passive transport in terms of your cells is when molecules are moving in and out without using any energy at all. And an active, actually, let me backtrack. Some of these molecules that can easily get in and out of your cell without using any energy are things like your oxygen molecules, carbon dioxide molecules, water, glucose, even types of waste. They are so tiny that they can easily get in and out of your cell without using any energy. And an active transport, like I was just talking about, that's when there's these molecules that are super big and they have trouble getting in and out of the cells easily. And these are things that your cells need, but they've got to use a lot of energy to get those big molecules in and out of the cell. Okay. There are two types of passive transport, and that's what you are going to be experimenting with this week with your gummy bears. So a type of passive transport is diffusion. So that's where molecules are moving. So for example, like water molecules are moving in an area of higher concentration to an area of lower concentration. So when I talk about higher concentration, think about an area that's like super crowded. And then an area of lower concentration is where it's not crowded. So osmosis is another type of passive transport. So we've got diffusion and osmosis. That's the lab you're doing. It's where water molecules, so osmosis, think water. It is where water molecules are moving from a higher area of concentration to a lower, and they're going through what is known as a semi-permeable membrane. So if you break that big word down, semi-permeable, semi means part of or partial, and then permeable means something where it's going through. Like if I just ate a piece of pizza for lunch and you were to come into my kitchen and you didn't know what I had, you probably could say, you know what, it smells like pizza in here, because it permeated, the smell of the pizza permeated through my kitchen, it spread around. So semi-permeable means something that can partially get through something. And this is another example of like a semi-permeable membrane. If you've ever made pasta before and you have a strainer, you want to keep the pasta, the good stuff that you're cooking, but you want to strain, you want to get rid of all that pasta water that you don't want to eat. So the actual strainer here is like the semi-permeable membrane. That's only holding on to what you want, and it's getting rid of all the stuff you don't need. All right, so another example, kind of like what's going to happen in your lab, is that you're going to have a beaker or like a clear cup, and these are all of our water molecules kind of bouncing around like crazy. So you've got H2O molecules, you got your O, your oxygen, which is the red bubble, and then the two hydrogens that are on the outside that look like little Mickey Mouse ears. They're bouncing around in this beaker. If you were to add food coloring to this beaker, so you have a cup of water, and you're going to add, let's say, green food coloring to this beaker. The green food coloring is going to immediately, you put one drop in or a couple drops, is going to sink to one area of the beaker, typically the bottom. And that's an area of high concentration right there. You've got all your green dye molecules sticking together. And then over time, they are going to permeate through this beaker of water. And it's going to spread throughout until the entire beaker of water is green. So that is diffusion right there. That's an example of passive transport. So osmosis, like I said, you think of osmosis, think of water. And you have this semi-permeable membrane. So pretend this is our cell right here, this blue circle, that's our cell. And then we have this red dotted line going around, that's our semi-permeable membrane. Inside, we only have 3 very lonely water molecules. And then on the outside, we have all of our crowded and our high concentrated area, all of our other water molecules. And they're trying to get into that cell. They're fighting to get in there. That is osmosis, an area of high concentration trying to get into a lower concentration. So they're super crowded. They don't want to be around each other anymore. They want to get into an area and spread out into that cell. And there you go. They get in, some of them get into that semi-permeable membrane. They're now in the cell. So now what your cells are doing, your body knows how to do this, and you don't even know what's happening. Your cells want to be at a balanced state. Your body wants to be at a balanced state. When it's unbalanced, that's when like you get sick, or when something is off in your body, that's when it's unbalanced. Your body constantly wants to reach something known as an equilibrium. Think equal, even. Your cells are working so hard to be balanced and to reach that equilibrium. So another example of diffusion that your body is going through, again, without you even knowing it, is when you're breathing. Your lungs are taking in oxygen when you inhale, and then they're getting rid of the waste of what they don't need. Same thing with plants. Our plants, without plants, we would all be dead because they're giving us our oxygen that we need. And a plant is taking in carbon dioxide. This is an example of diffusion. So I'm going to kind of speed ahead a couple slides just because we don't need to stick on this too much. Like I said, I just want the concept to kind of sink in. Same thing in terms of like a fish. When the concentration of oxygen molecules is higher in the water than inside the actual like shark's body, oxygen is going to enter through its gills and move into a lower area, into its bloodstream. So this organism is at a balanced state. It's getting what it needs and getting rid of what it doesn't need. Same thing when you eat. Maybe you guys are hungry right now, and after consuming water or food, it is being broken down through your digestive system, and there is a higher area of concentration now in your digestive system than in your actual bloodstream. And as the water and even your food is moving through the tissues of your body and the cells, it is then getting into the bloodstream so it can pump through your body, and that's where your body is getting its energy, what it needs. Sorry, I'm just skipping through the next few slides. You can always go back to it, but the reason that diffusion and osmosis are so important in living things is, like I've been trying to really highlight, is that your body needs materials. Your cells need these things that are going in and out of the body all the time. Otherwise, any living organism would not survive. So in this example, this is going to show you what active transport is. And I'm not going to spend too much time on this because you're doing a lab on passive transport. But just to get an idea of what active transport is, these big red molecules here are an example of what this is a plant cell. We know it's a plant cell not only because it's green, but because it's rectangular in shape. And we have our semi-permeable membrane that's going around there. But you can notice these little openings here, they're not that big, but somehow these big red molecules need to get in here. They need to get through that. They need to squeeze through that little tiny spot of the cell membrane. in order to do this, your cells or the plant cells need to work super hard for this to happen. So there is a process called endo and exocytosis. So if you think of the word or the prefix endo, that means in, and then the same thing, exo means out. So your cells are actually opening up and they're engulfing the particle that it needs to take in. And then once your cells are getting what they need, they're kind of like, I'm done with you. I'm going to spit you out now. They need to open up again and push out the waste. But these molecules are so big that the cells need to use energy to do this. And that's basically what endo and exocytosis is. It's active transport. Okay. I posted a video here. It's a lab I wanted to do with you, but we weren't able to provide you with eggs, and I didn't want you to have to take any eggs from your kitchen that you would be using for like breakfast or dinner. So, but if you want to do this lab, even like after our class is done, like after the summer course, let me know. I can send you directions on how to do it, or you can easily look it up on YouTube directions. But I'm going to play this video because I think it's pretty cool to show you an example of passive transport. Okay, so that lab is actually showing you like that actually happens to an egg when you put it in. Cairo syrup is just like corn syrup, or you could use honey, or even something like molasses. It's like that thick syrup, as opposed to like water or vinegar. But it's actually the process of osmosis and diffusion. So you saw the egg like shrunk, and then the other egg got like really squishy and maybe even a little bigger. And it actually bounces. Like you could actually bounce it on the ground. Like it's not going to break open like a raw egg. It's a really cool experiment to do. You really don't need a lot of materials, but again, I didn't provide you, I wasn't able to provide you with the egg. So I just thought it was something cool to show you. But now getting to the lab that we are going to do, I'm going to kind of speed through these question and answers because this is something that you can go back into the slideshow. I provided the answers for you, so that should hopefully be helpful. But now to the fun experiment that I want you all to do this week. I wanted to give you a little bit of a science, some background information about gummy bears, these delicious little gummy candies that I love. I do want to mention, I don't want you eating during the lab. So any gummy bears that you are experimenting with, please do not eat. If you have like gummy bears, like a side that were left in the bag, feel free to eat them if you want. But whatever you are experimenting with, I can't say it enough, please do not eat them. It is for science only. But gummy bears are made of a solid jello-like substance known as gelatin and sugar. When gelatin, pretty much if you've ever made jello before, you're given this little packet of like powder and you mix it with warm water. You typically boil it. And then when it cools and you put it in the refrigerator, like overnight or a couple hours, it solidifies. And so basically gelatin is a protein known as collagen. And when it's heated and then cooled, the proteins actually form these chains and these bonds. So they like come together and they form and they become this like gelatinous like substance. And the cool thing, a very interesting fact that you might not know, is that your body is made up of these proteins. Like your whole body is made up of protein. But collagen is found in your bones and other tissues in your body. And that's actually one of the main things. I don't know if some of you are into like having like smoothies with like protein powder, or putting this in like your like energy drinks if you're into like working out. But collagen is what basically keeps your skin firm. And when you get older, you lack a lot of these proteins in your skin. So a lot of people take these supplements to keep their skin young and firm. So if you have anybody in your family that's like a grandparent or like an elderly neighbor or even like a teacher, you might notice some sagging skin, and that's because their collagen, they don't have a lot of it in their body anymore, and that's what happens. So the materials for the Gummy Bear Lab I have listed here. This was on your supply list and in your kit. You will need a bag of gummy bears, water, white vinegar, salt, baking soda. Sugar is actually optional. Is optional. And then 4 beakers. If you don't have the beakers, you can use clear plastic cups. Labels, you can use like a piece of tape if you don't have a label, and then a scale and a ruler are really important, especially the ruler above all. If you don't have the scale, that should be okay, but the ruler is important to make some measurements. And you will need a marker. I didn't list it on there for your label, so you can mark each of the beakers or the cups that you have set up. So the lab that you're going to be doing, let me pull up for you. Okay. So these are the measurements that you're going to have to be taking. I listed here in step D and F, you're going to be finding a simple calculation for volume, which is labeled as V, and then D is known as density. So with those 2 calculations, those are 2 things you really need in order to find out your accurate results from the lab. In part B, I go through step by step how to set up the lab. So you're going to need 4 cups, which I labeled here. You need water in 1 beaker, vinegar in another. You're going to need a partial mix of water and baking soda, and then water and salt. If you want to add a 5th cup, it's optional, but you can add sugar if you want. You can just use the same measurement, water with 15 milliliters or 1 tablespoon of sugar. So that's for day 1. Day 1 should not take you very long. You are going to be taking measurements and then setting up the cups. So that's day 1. And then on day 2, you are going to remove carefully the gummy bears from each mixture, and then you're going to kind of place them on a paper towel or even like a paper plate, let it dry, and then you're going to then make a second round of measurements after they have sat in the liquid mixture for about 24 hours. But be very careful when you take the gummy bear out, because they are going to be very delicate and I don't want them to break. If it does break, don't get upset and think you have to do that lab over again. Just try to kind of piece the gummy bear together, or have it all together in an area where you can make a measurement. This is the chart that you will document some of your observations. And then here is your experiment data chart. So you can see here you have day 1 and day 2 of your measurements and your calculations. And then, so you have here like for the water and salt, vinegar, and then water and baking soda. And then after you have all of your data to look at, recorded, look at the any amount of changes that happened, and then you're going to answer these discussion questions. I do have a bonus activity. If you want to do that, it's optional. And then here is the list where I would like you to, a chart, I should say, of the scientific method to help you kind of put together your observations and record what happened after you were done. I will have that posted for you at some point today after class. And I told the other classes I typically would want your assignments in by midnight on Sunday, but because it is a 2-day lab experiment, I'm going to give you an extra day. So I'm going to give you to the end of the day on Monday. Monday at midnight, you have to do the experiment, take your pictures, videos, finish your lab report, and upload it to me by the end of the day on Monday. So.
Can I say something really quick?
Yeah.
So I don't know if you guys heard Miss Amy during the morning announcement, but in Group C, only 1 or 2 people actually submitted their past assignments, and Miss Nicole is still accepting late assignments. So please do them, or we might have to release you from the program if you're not gonna do any work. So this is just a FYI to everybody. Like you're not just gonna sit here and do nothing and expect to earn the full stipend at the end of the summer. So, yeah.
And we're here to help you guys. So if you have questions, if you feel like you have a lot on your plate right now and you just have trouble getting some of it in, reach out to us, because if we see that you're trying and you're doing the work and maybe you're struggling in some areas, that's fine. We just want to see that you're trying and
Right.
putting forth some type of effort.
Yeah.
You know, I do have office hours tomorrow. But aside from office hours, if you have any questions or need help with anything, please reach out to Miss Emily, Miss Athena, myself. We're all here to help you. And just on a final note, on my module for week 3, not only will I have the lab posted for you, I will post today's class that I've recorded it, so that will be up if you want to go back to it. If you feel like you missed something or you want to go back to review it. But also there's a movie that's posted. It's optional. You don't have to watch it, but I love the movie. It's an older movie, and Miss Athena has also seen it. She actually came up with the idea to post it because she liked it so much. So it's called Osmosis Jones. Like I said, it's an older movie, but it's pretty funny. It's a little wacky, but it goes over the concept of osmosis. And it was an actual movie that, believe it or not, was in the movie theaters. I think Chris Rock we were trying to talk about that. I think he's one of the voices, the characters in the movie. So I think you guys would really like it. I love it. I've shown the movie so many times. So it's up in the module. The first half of it's posted. And then next week we'll upload the second half, because it's about like an hour and 25 minutes, I think. So feel free to watch it if you're bored and you don't want to be outside because it's so hot. Check out the movie. It's in the module. So we have about maybe 2 minutes before we say goodbye. So any last questions? Let me check into the chat because I haven't done that in a minute. But you've seen it.
So
Okay.
it's a 2-day lab.
Yes.
Oh, and on the I thought I seen like more days on the thing that you showed us.
What do you
Like
mean more days?
Like you said, like it had like a chart. It said like day 1, day 2. Day
Yeah,
3.
it's just day 1 and day 2. So for day
Okay.
1, you're basically going to be taking your 4 gummy bears out of the bag, and you're going to be doing some measurements that you're recording. And then you're going to set up your 4 beakers, and you're going to place each gummy bear in a different beaker. and have them labeled so you know which one is what, because you're going to have one with just water, one with vinegar, baking soda, and salt. And then you're going to let 24 hours go by. And so then on day 2, you're going to gradually take the gummy bear out of each beaker, and then you're going to continue doing the same exact measurements that you did on day 1. And then you're going to add that to your day 2 chart. And then that's when you're going to make your observations and answer the questions.
And are like, cause like it's like kids in here, so like we don't really have any like glass or plastic clear cups. We just have like colored cups and stuff like that.
That's
So
fine.
would it be cool if I put in like a wine glass or something?
Any glass you can use, but just make sure that you have 4 of the same glasses.
Okay.
So
But
you
they
want to make sure that you have the same
possibly,
amount of liquid in each glass. You don't want it to be like different, because then your results won't be accurate.
would they be able to use
Okay.
like empty water bottles?
That water bottle might be hard because of the opening isn't that large. So it would be hard to get the gummy bear back out of it without.
I mean, they could probably cut it, I think, or something like that, you know?
Yeah, I mean, if you have to use a water bottle, that's fine as like a last resort. I would recommend using, like here, this is just my coffee glass, but like a glass like this would be, if you could find 4 glasses, or if you have even like a solo cup, even if it's like not clear, if it's like one of the red solo cups or another cup you may have, like a plastic cup, that's fine.
You guys
Ideally,
are provided
I
with
would like it to
beakers.
be a clear cup, but
Just so you know, you guys are provided with beakers in your summer supply box.
yeah, I thought they were in there, so you should have them. But
There are 4 beakers, labels, ruler, sugar, baking soda, salt, and a bag of gummies for this lab. That is provided to you. The only thing that is not provided to you is water and white vinegar for this lab. So everything else you guys should have.
okay, good. That's what I thought. And like I said, the sugar is optional, so it's not even listed on my lab report. That's something that you can do as like a 5th cup. That's up to you. Okay. But other than that, if you guys still have any questions or anything, please reach out to us. Otherwise, class is over.
Seniors are coming in.
Big pictures.
Okay, and wait, and we can leave it out, or do we have to put in like the refrigerator or something?
Oh no, you can leave it out. Room temperature.
Okay.
You can leave it out on the counter.
Okay.
But
Big
leave it in an
picture.
area
Right.
that is not going to be tampered with. Like if you have like a pet or a sibling, you don't want it to be knocked over.
Okay.
All right. Bye, guys.
Bye, Lamar.